Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's think about this. If you are one of the millions, and I mean millions, of people who have consciously, swapped traditional sugar for a low-calorie alternative, maybe it's to manage your weight or get better control over blood sugar, or maybe just aiming for a generally healthier lifestyle. Well, you've made a strategic choice, right, to protect your long-term health. But what if that choice, that seemingly harmless powder or liquid you add to your coffee or find in your protein bar, what if it was quietly undermining your brain's most vital defense system, and at the same time, actually increasing your measurable risk of stroke?
That is the really unexpected and frankly quite concerning question that's being raised by some recent and pretty comprehensive research. A lot of it's coming out of the Integrated Vascular Biology Laboratory at the University of Colorado. So today we're doing a deep dive into erythritol. It's a sugar alcohol, and it has become, well, absolutely everywhere in the modern health food landscape. You find it in protein bars, energy drinks, loads of baked goods, and especially anything labeled sugar-free or keto-friendly.
Okay, let's unpack that straight away, because erythritol seems to occupy this kind of unique spot in the world of sweeteners, doesn't it? It's a sugar alcohol, a polyol. What makes it the go-to choice for manufacturers compared to maybe older, more controversial ones like aspartame or sucralose?
Well, the huge popularity really comes down to its functionality and, importantly, its taste. It mimics sugar really quite closely. It's roughly, say, 60 to 80% as sweet as regular table sugar. But critically, unlike some other sweeteners, it generally doesn't have that sort of bitter or metallic aftertaste that often screams artificial to people. And for the health-conscious consumer, the calorie count is tiny, just about more 0.2 calories per gram. Practically nothing.
And because it tastes okay and has almost no calories, it's saturated the diet industry. You see it on the ingredients list of pretty much everything trying to hit that low-carb, zero-sugar target. It's marketed as, safe, natural, even healthy.
Exactly. And importantly, its classification as a sugar alcohol has given it a really significant regulatory advantage over the years. Regulatory bodies pretty much worldwide, like the FDA in the US and the European Food Standards Agency, they've long considered it safe for consumption. It's also managed to mostly avoid the kind of intense scrutiny that's been applied to truly artificial sweeteners. So, for example, it wasn't specifically called out in those recent World Health Organization guidelines that cautioned against using other non-nutritive sweeteners for long-term weight management.
The sources we looked at also mentioned something manufacturers often point to sort of bolster its safety image. The fact that our bodies actually produce erythritol naturally, endogenously, in really small amounts when we metabolize glucose and fructose, it gives it the sort of veneer of being natural and compatible with our system.
That perceived naturalness is definitely part of the appeal, maybe even part of the marketing. However, the research we're really focusing on today, it completely reframes that assumption. Our mission here is to explore these findings, particularly from the University of Colorado team. They went beyond just showing a correlation. They seem to have established a direct mechanism of harm. They've provided some pretty troubling new details that link consuming erythritol, even acutely, directly to how our vascular system functions, especially those protective structures in the brain. And this reinforces the clinical risk of cardiovascular events that earlier studies had hinted at.
Right, and this is where we pivot from, theoretical risk to actual documented risk in people. Before these researchers even started their cell experiments, there was already some, disturbing real-world epidemiological evidence piling up. We need to set the stage here. What did those earlier large studies suggest, and why did they make scientists dig deeper?
Yeah, the initial clinical picture wasn't exactly subtle. This new research didn't just appear out of nowhere. It was definitely spurred on by some alarming data from large, long-term cohort studies, reckon studies involving tens of thousands of participants, followed over many years. And these studies had repeatedly flagged a statistically significant link between having high levels of erythritol circulating in the blood and experiencing serious adverse cardiovascular outcomes.
Okay, so not a small patient group. These are massive, long-term population studies showing this connection. What specific kinds of events were elevated? What were they seeing?
The main risks centered on what clinicians call major adverse cardiac events, or ME. That's basically the big three: a non-fatal heart attack, a non-fatal stroke, or dying from a cardiovascular cause. The analysis really honed in on people whose blood plasma levels of erythritol were in the highest quartile. So the top 25% of the population studied, the ones with the most erythritol in their system.
And what did the data show for that top quartile. What was the magnitude of this association?
The finding was pretty stark enough to really prompt this deeper mechanistic investigation. Participants with the highest blood levels of erythritol were found to be approximately twice as likely, double the risk to experience a major cardiac event, like a heart attack or particularly an ischemic stroke, compared to the people with the lowest levels.
Twice as likely. Wow, that's a huge multiplier. And it's really concerning to see that tied to something marketed as a harmless food additive. Was this risk just concentrated in people who were already quite sick, maybe with uncontrolled diabetes or heart disease?
That's a really crucial point, and the observational studies specifically controlled for that. The sources emphasize that this association, this doubled risk, remained strong and statistically significant, even after accounting for other traditional cardiometabolic risk factors. So they adjusted the data for things like high blood pressure, high cholesterol, existing diabetes, age, smoking history, all the usual suspects. And even then, high erythritol levels still stood out as an independent factor associated with increased risk.
Okay, that completely shifts the conversation. It's not just erythritol might be risky if you're already unwell.
Yeah.
It becomes erythritol might pose an independent vascular risk to almost anyone who consumes it regularly.
Precisely. But, as compelling as those epidemiological links were, they could only establish a correlation, an association. They showed that people with high erythritol tended to have more heart attacks and strokes. Some studies even suggested it might increase thrombotic potential, basically, making blood platelets stickier and more prone to clotting.
But science demands more than just correlation, right? They needed the smoking gun, the actual mechanism. How exactly was erythritol potentially going from being a sweetener in your yogurt to actually causing damage at the cellular level in critical blood vessels, especially those feeding the brain?
And that's exactly what pushed the University of Colorado team to shift gears, moving from looking at large populations to focusing on fundamental cell biology. They needed to see if erythritol was directly harming the blood vessel cells themselves, causing them to malfunction. That would provide the missing piece, the mechanistic explanation for that observed doubled risk of stroke and heart attack.
Which brings us directly to the system most vulnerable when blood vessels are compromised. The brain's ultimate security checkpoint. Let's dig into the specific target of this research. We talk about the blood-brain barrier almost like it's an abstract idea, but it's a real physical cellular structure. Can you explain why it's so absolutely critical for brain health and what happens when it's compromised?
Yeah, the blood-brain barrier, often called the BBB, is arguably one of the most critical protective systems we have. You can think of it less like just a border crossing and more like a highly selective, heavily fortified wall around the brain. It's mostly made up of specialized cells called microvascular endothelial cells. These cells line all the tiny capillaries, the smallest blood vessels, inside the brain. And they're joined together by extremely tight connections called tight junctions. They form a physical seal, a barrier that's meant to be very difficult to cross.
So it's not just filtering things, it's a physical gatekeeper, being very strict about what gets in and out.
Exactly. Its job is really twofold. First, it has to strictly control which specific substances, mostly essential nutrients like glucose and amino acids, are actively transported into the delicate brain tissue. And second, its job is to rigorously keep out harmful stuff, things like toxins, pathogens, even immune cells from the rest of the body that could cause inflammation or direct damage to neurons. If those tight junctions weaken, even a little bit, the brain becomes vulnerable.
Okay, so given how vital that barrier is, the Colorado study aimed specifically to isolate what effect erythritol has on these particular brain endothelial cells, their structure and their function.
Yes, precisely. They worked in vitro, which just means in a lab dish outside of a living organism. They cultured human cerebral microvascular endothelial cells, these key BBB cells, so they could study them in isolation. This way, they could be more confident that any effect they saw was directly due to the erythritol itself and not influenced by other complex factors happening in the body.
Can you walk us through the methodology? How did they decide on the dose of erythritol? And importantly, what does that dose actually represent in terms of real-world consumption?
Okay, so the researchers exposed these human brain endothelial cells to a single acute dose of erythritol. The concentration they used was 6 millimolars, or 6 milliriemer. Now, that number wasn't just pulled out of thin air. It represents a really key physiological level. They calculated that this 6 millimolar concentration is basically the average level of erythritol found circulating in a person's blood after they consume a single standard-sized product sweetened with a typical amount of erythritol, think around 30 grams.
Right, and that's a critical point because it moves this from some abstract lab finding to something directly relevant to public health. We're not talking about some massive toxic dose nobody would ever encounter. We're talking about the concentration your blood might reach after, say, drinking one bottle of a zero-sugar sports drink or eating one of those popular keto protein bars.
Exactly. It mimics a pretty common everyday exposure scenario.
Yeah.
And, if you're having a couple of those products spread throughout the day, your blood concentration could potentially remain quite high for extended periods. So based on what's already known about how blood vessels can become dysfunctional, the researchers had a specific hypothesis about what this dose would do. They expected to see basically three things. First, an increase in cellular stress. Second, problems with blood flow regulation. And third, a weakening of the body's natural clot-dissolving system.
So they set up specific markers to look for: increased oxidative stress, reduced nitric oxide levels, promotion of a substance called endothelin-1, and problems with the release of a key clot-buster protein, tissue-type plasminogen activator, or TPA. This really sets the stage for seeing cellular malfunction across several critical pathways that influence stroke risk.
And the results they got were, well, pretty clear-cut. The cells, even studied in isolation like this, showed significant signs of dysfunction in response to this single clinically relevant dose of erythritol.
Okay, this is where we really get into the nitty-gritty, the mechanism, the biological why behind the epidemiological what. Let's start with the first major finding, that immediate and sort of universal type of cell damage. They observed oxidative stress.
Right. So when any cell experiences stress, maybe from an infection or a chemical toxin, it starts generating these highly reactive molecules called reactive oxygen species, or ROS. Most people know them as free radicals. They're unstable molecules, chemically speaking, and they want to react with anything nearby: DNA, cell membranes, proteins causing damage. It's like molecular wear and tear. And what the study found was that erythritol triggered a significant surge in the production of these ROS inside the brain endothelial cells.
How significant was this burst of damaging molecules?
They measured it. ROS production was approximately 75% higher in the brain endothelial cells that were treated with that 6 millimolar dose of erythritol compared to the control cells that didn't get any erythritol. That's not a small fluctuation. It indicates a state of real stress for the cell. This sudden, large burst of free radicals poses an immediate threat to the integrity of the cell itself. And crucially, for the blood-brain barrier, it threatens the tight junctions that seal the barrier.
So by causing this huge wave of oxidative stress, erythritol is essentially poking holes, metaphorically speaking, in the brain's protective wall. Even if the cell doesn't die right away, the barrier's integrity is compromised.
That seems to be the implication, yes. Now, the cells do try to fight back. The paper noted that the cells showed what's called a compensatory defense response. They actually started producing more of their own internal antioxidant proteins, things like superoxide dismutase 1 or SOD1 and catalase. These are the cell's natural scavengers trying to neutralize those free radicals.
But the fact that the overall ROS level still ended up 75% higher suggests that defense just wasn't enough. It got overwhelmed.
It appears to have been completely insufficient to negate the initial onslaught. The fact that the cells ramped up their antioxidant production actually just confirms how severe the initial chemical stress from the erythritol was. The net result was still significant dysfunction, indicating the erythritol-induced stress overwhelmed the cell's ability to maintain balance, or homeostasis.
Okay, so finding one. Acute widespread cellular damage via oxidative stress, compromising the security system. Now let's move to finding 2, which is about controlling blood flow itself, vasomotor regulation. And this depends on a really delicate balance, a push-pull system.
Indeed. Healthy blood vessels, especially the tiny ones in the brain, are constantly adjusting. They act like very sensitive traffic controllers for blood flow. They need to dilate or relax and widen when the brain needs more oxygen, maybe during intense thought or stress. And they need to constrict or tighten when less blood flow is needed. This crucial balance really hinges on 2 key signaling molecules: the main relaxer molecule, which is nitric oxide, or NO, and the main constrictor molecule, endothelin-1, or ET1.
So what did the erythritol do to the green light signal, the nitric oxide that tells vessels to relax?
It severely hampered it. The study found that erythritol significantly reduced nitric oxide production by about 20% compared to the untreated cells. Now, a 20% drop in NO production might not sound like everything stopped, but in vascular biology, that's a serious disruption. Nitric oxide is this constant protective signal that helps keep blood vessels flexible, open, and healthy.
So it wasn't like the cells ran out of the building blocks for NO. It was more like the factory producing it got sabotaged. If the endothelial cells are the NO factory, how exactly did erythritol interfere with the production line?
Yes, that's a good analogy. And this is where the study gets quite specific about the mechanism. Nitric oxide production relies heavily on a key enzyme called endothelial nitric oxide synthase, or eNOS for short. The activity of this eNOS enzyme isn't just on or off. It's very precisely controlled by adding or removing chemical tags called phosphate groups, a process called phosphorylation. Think of these phosphate tags as on-off switches or dimmer switches. What the study showed is that erythritol messed with this phosphorylation pattern in a way that effectively turned down the activity of the eNOS enzyme.
The researchers looked at two specific switches, two phosphorylation sites on the eNOS enzyme. Can you break down what those sites do and how erythritol flipped them the wrong way?
Sure. The two key sites they looked at are known by their positions on the enzyme, serine 1177 and threonine 495. When a phosphate group gets attached to serine 1177, that's the on switch. It strongly activates eNOS, leading to lots of nitric oxide production and vessel relaxation. Conversely, when a phosphate gets attached to threonine 495, that's like an off switch or a brake. It inhibits eNOS activity, reducing NO production.
Okay, so it's a sophisticated system designed to fine-tune blood flow based on the brain's needs. What exactly did erythritol do to these crucial switches?
The study demonstrated that in the cells treated with erythritol, the phosphorylation, the on signal at that activating serine 1177 site was significantly lower. At the same time, the phosphorylation, the off signal at the inhibitory threonine 495 site was significantly higher.
Wow, so erythritol managed to simultaneously take the foot off the accelerator, less activation at SERTL 177, while also slamming on the brakes, more inhibition at 325. That directly explains the 20% drop in nitric oxide. The vessel can't relax properly because its internal signaling is actively being suppressed.
Exactly. And that's only half the picture of the imbalance. Now we need to look at the other side, the constrictor molecule. What happened with endothelin-1?
It went the other way, I presume, promoting constriction?
Precisely. Endothelin-1, or ET1, is known to be one of the most potent and long-lasting vasoconstrictors the body makes. What the study found was that erythritol caused a significant increase in the precursor molecule, called big ET1, which then led to endothelin-1 production itself being boosted by about 30% in the treated cells compared to controls.
Okay, let's put that together. A 20% reduction in the primary relaxer, NO, and a 30% increase in the primary constrictor, ET1. That represents a massive shift towards a constricted state in these critical brain blood vessels. What does that dual action mean clinically for the brain?
It results in blood vessels that are sort of chronically biased towards being tight and constricted. Researchers refer to this as creating a pathologic vascular phenotype. This combination, low nitric oxide availability coupled with high endothelin-1 production, is a well-recognized warning sign for increased risk of ischemic stroke. The vessels become stiffer, narrower, less responsive. This reduces the brain's ability to get the oxygenated blood it needs, especially during moments of demand, and makes it much more vulnerable if there's any kind of blockage or drop in blood pressure.
Which brings us to the third major finding, and this feels like where the immediate danger really ramps up, the impact on the body's ability to actually clear a blockage once it starts to form.
Yes, this is sort of the double whammy effect that seems to be emerging. The earlier observational studies hinted that erythritol might make blood more likely to clot, increase thrombotic potential. This cellular study found that it also cripples the body's ability to dissolve those clots once they form, impaired fibrinolytic capacity.
Can you explain the body's natural clot-busting defense? How does that normally work?
Healthy endothelial cells, the same cells lining the blood vessels, are constantly working to prevent inappropriate clotting. But if a clot does start to form, maybe due to that increased platelet stickiness, these endothelial cells are supposed to respond immediately by releasing a crucial protein called tissue plasminogen activator, or TPA. TPA's job is to actively break down fibrin, which is the protein mesh that forms the structure of a blood clot. Think of TPA as the body's own emergency response system to dissolve blockages, especially in the brain, before they cause major damage like a stroke.
So the researchers tested this response. They stimulated the cells in the lab to mimic a situation where a clot is forming. What happened to TPA release in the cells exposed to erythritol?
They found that erythritol significantly blunted the release of TPA in response to that clotting stimulus. It's interesting, the baseline sort of background level of TPA release wasn't really affected, but that critical rapid surge of TPA that's needed to fight off a developing clot, that response was severely impaired in the erythritol-treated cells. They couldn't mount the defense when it was most needed.
So let me get this straight. Erythritol seems to make the vessels tighter and potentially makes the blood stickier, and it disables the body's fire extinguisher right when the fire starts.
That appears to be the devastating combination suggested by these findings. This impaired fibrinolytic capacity means that if a small clot does form, perhaps initiated by the increased platelet reactivity seen in earlier studies, it's far more likely to stick around, grow, and potentially lead to a major debilitating stroke. The overall picture from these cellular mechanisms, the oxidative stress, the shift towards vasoconstriction, and the failure of clot dissolution, paints a really comprehensive and concerning picture of how erythritol might sabotage cerebrovascular health.
That is a tremendously detailed cascade of negative effects at the cellular level. So just to synthesize this section clearly, a single dose of erythritol at a concentration people commonly reach after one sweetened product caused a 75% spike in damaging oxidative stress, significantly messed up the balance controlling blood vessel relaxation and constriction, and fundamentally compromised the body's vital clot-busting system.
Absolutely. And having laid out that detailed mechanism, the potential implications for public health become pretty hard to ignore. These in vitro findings, showing effects on specific molecular pathways, provide that crucial mechanistic support that was missing from the earlier observational studies. They help validate why those studies might have seen that doubled risk of heart attack and stroke. We're moving beyond just statistics now. We have a potential biological explanation.
And the specific things measured the problems with ENOS activation, the high endothelin 1, the poor TPA response. These aren't just obscure lab markers, right? These are established indicators of cerebrovascular dysfunction. Doctors look at these kinds of imbalances when assessing stroke risk in actual patients. It seems erythritol is directly inducing a known high-risk state in the brain's delicate blood vessels.
Precisely. The research strongly suggests that this sweetener, which is everywhere, profoundly disrupts the normal, healthy functioning, the homeostasis of these critical brain microvascular endothelial cells. This understanding really demands a shift, I think, in how regulatory bodies, healthcare providers, and consumers view this product. The long-held assumption of safety, based mainly on its low calorie count and the fact it occurs naturally in tiny amounts, is now being seriously challenged by this mechanistic evidence.
But of course, in the spirit of good science, we have to acknowledge the limitations here. This was research done on isolated cells in a lab dish. What are the important caveats we need to keep in mind when interpreting these powerful findings?
That's a critical point. The primary limitation, which the University of Colorado scientists themselves are very clear about, is the in vitro setting. Cells grown in isolation on plastic bathe constantly in a precise chemical concentration. Well, that doesn't perfectly replicate the incredibly complex environment of the human circulatory system.
Right. In a living person, that erythritol has to be absorbed, travel through the bloodstream, maybe get processed by the liver, interact with immune cells, deal with fluctuating blood pressure, hormones, all sorts of other factors.
Exactly. There are countless other biological factors in a living system that might potentially buffer the effects seen in the dish, or conversely, they might even amplify them. We don't know for sure yet. The researchers themselves strongly recommend that the next essential step is more sophisticated testing. For instance, using advanced organ-on-a-chip, or specifically blood vessel-on-a-chip systems. These are microfluidic devices that can better mimic the physical forces, like blood flow, pressure, and the interactions between different cell types found in actual human blood vessels. Getting similar results in those systems would provide much stronger confirmation.
And there's also the question of timing, isn't there? They used a single acute dose, like drinking one beverage, that gives us a snapshot of the immediate hit. But what about the way most people consume these things, which is often chronically, day after day?
Yes, that's probably the biggest unanswered question right now, and it's a major area of concern. Many frequent users of these products, maybe people managing diabetes very strictly, or those on long-term ketogenic diets, they might be consuming non-nutritive sweeteners multiple times every single day. This likely means their blood levels of erythritol aren't just spiking briefly after one product, but could be staying persistently elevated for much of the day, day in and day out.
And what might that persistent elevation mean based on what we've learned about the damage from a single acute hit?
Well, we know from this study that each acute exposure seems to cause significant oxidative stress and throws off that crucial nitric oxide endothelin 1 balance. If that's happening repeatedly, constantly, you can imagine the cumulative toll on the endothelial cells. These cells are always working, repairing themselves, regenerating. But facing that kind of stress day after day, year after year, it could plausibly lead to chronic damage. Maybe permanent stiffening of the vessels, gradual breakdown of the blood-brain barrier integrity, essentially accelerating the processes that underlie conditions like atherosclerosis and increasing the risk of long-term cerebrovascular disease. The chronic effects of constantly bathing the vascular system in high concentrations of external erythritol are really an urgent question for future research.
This really forces us into a difficult conversation about a major public health trade-off, doesn't it? Sweeteners like erythritol were initially embraced and are still promoted as a solution to a huge problem: the health consequences of excessive sugar consumption, like obesity and type 2 diabetes.
That really is the crux of the dilemma. Experts in metabolic health, like Professor Havovi Chichgar, who is cited in the materials, quite rightly point out that low-calorie sweeteners, including erythritol, have been valuable tools. They genuinely do help many people reduce their overall calorie intake and, importantly, manage their blood glucose levels. And controlling blood sugar is absolutely critical for preventing the devastating long-term vascular damage caused by uncontrolled diabetes.
So it's this difficult balance. On one side, you have the very well-known severe long-term risks associated with eating too much sugar, risks that fuel global epidemics of obesity, diabetes, and related cardiovascular disease. But now, on the other side, we have compelling mechanistic evidence suggesting that a popular substitute, erythritol, might potentially weaken the brain's defenses and acutely increase the risk factors for stroke and heart attack.
The conflict is becoming much clearer, and it leaves consumers in a really tough spot. Are you potentially swapping a known chronic metabolic danger from sugar for a possibly more immediate acute vascular risk from the substitute? This research really underscores a broader challenge in nutritional science, which is understanding the true, long-term, systemic effects of food additives that become incredibly widespread in our diets relatively quickly, often before all the potential biological consequences are fully understood, especially concerning something as fundamental as vascular function.
Yeah, it feels like we're often playing catch-up. We introduce a substitute to solve one problem, and then years down the line, we start identifying potential unintended consequences. This study feels like a significant moment, maybe demanding a pause and more critical look at every sugar-free label we see.
So to kind of wrap up the key findings from our deep dive today, this research from the University of Colorado provides pretty definitive mechanistic data suggesting erythritol isn't just a passive, inert substance passing through your body. At concentrations that are easily reached after consuming just a single typical sweetened product, it seems to actively promote a specific vascular phenotype known to be associated with higher risk.
And we now have a clearer picture of how it might be doing that. Through that triple threat mechanism, the big 75% surge in oxidative stress damaging the vessel lining, the significant disruption of the nitric oxide endothelin 1 balance pushing vessels towards constriction, and the worrying impairment of the body's natural clot-dissolving TPA system.
So for you listening to this, the main takeaway is perhaps a clearer understanding of the potential risk profile. While erythritol definitely helps people avoid the calories and glucose spikes from sugar, frequent or high-dose consumption might, over time, be subtly compromising the health of your vascular system, including the critical protection of your brain. It really does compel us to reconsider our reliance on any kind of substitute additive, even those that seem natural or have regulatory approval, and maybe ask tougher questions about whether the perceived benefit of substitution always outweighs any potential hidden costs.
And that brings us to our final provocative thought for you to mull over after this deep dive. We mentioned earlier that erythritol is actually produced naturally, endogenously by our own bodies, but in very small amounts. Our bodies are presumably equipped to handle that low level of internal production. So what does this new data showing problems at much higher concentrations from external sources suggest about the potential difference, maybe even danger, of consuming this exogenous, this added erythritol, especially at these massive acute spikes that are far beyond anything the body would naturally encounter on its own? What might be the long-term consequences of constantly pushing that system way past its normal, evolved operating range?
That's really the fundamental question, isn't it? Is the body's baseline ability to manage tiny amounts of its own erythritol fundamentally different from the systemic impact of flooding it with high concentrations from processed foods and drinks? Until we get more data, perhaps from those more sophisticated vessel-on-a-chip studies or long-term human trials that can provide clearer answers, Perhaps minimizing the intake of any highly concentrated sweetener might just be the most prudent approach for protecting long-term vascular health.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.